In the video, we will explore the intricate process of embryonic gut development. We will cover the formation of the gut tube, the folding and rotation of the gut, and the development of the gastrointestinal tract. We will also discuss the development of the enteric nervous system, gut-associated lymphoid tissue, gut epithelium, pancreatic bud, liver and biliary system, mesenteries, intestinal villi and crypts, Meckel's diverticulum, and the anal canal. In the early stages of embryonic development, cells in the embryonic disc fold inwards to form three germ layers. The endoderm layer gives rise to the primitive gut tube, which develops into the gastrointestinal tract. The gut tube is initially divided into foregut, midgut, and hindgut, which undergo further specialization. Researchers continue to investigate the mechanisms behind this process. In this video section, we will explore the complex processes involved in the folding, elongation, and fixation of the gut during embryonic development. These processes lead to the final anatomical structure of the gastrointestinal tract. Researchers are currently investigating the specific molecular signals and mechanical forces that drive these intricate changes. The gastrointestinal tract develops into the esophagus, stomach, small intestine, and large intestine. The foregut, midgut, and hindgut give rise to these divisions, which then undergo morphological changes and differentiation to form specialized tissues and organs. Accessory digestive organs such as the liver, gallbladder, and pancreas also develop and contribute to digestion, nutrient absorption, and metabolic regulation. The enteric nervous system, ENS, develops from neural crest cells and regulates gastrointestinal function. It controls motility and secretion, coordinating muscle contractions and glandular secretions for digestion. Understanding ENS development is crucial for preventing gastrointestinal disorders. The gut-associated lymphoid tissue, GALT, protects the gastrointestinal tract from harmful invaders. It includes structures like Peyer's patches, lymphoid follicles, and isolated lymphoid follicles that house important immune cells and develop through interactions between different cell types, ensuring effective immune monitoring and targeted immune responses. The development of the gastrointestinal tract involves a complex process of gut epithelium differentiation, leading to specialized cell types like absorptive cells, goblet cells, and enteroendocrine cells. These cells have important roles in nutrient uptake, protection of the gut, and hormone production for digestion. Precise signaling pathways and gene regulatory networks orchestrate their development. This knowledge is crucial for medical students in understanding the digestive system and diagnosing and treating related disorders. The pancreas, an essential organ for digestion and metabolism, develops from two progenitor structures, the ventral and dorsal pancreatic buds. These buds emerge from the foregut endoderm and undergo complex morphological changes before ultimately fusing together. The proper budding, rotation, and fusion of the ventral and dorsal pancreatic buds are orchestrated by specific genetic and molecular cues. Disruptions in this process can lead to pancreatic malformations and impaired functionality. The liver, gallbladder, and bile ducts develop from the hepatic diverticulum. This process involves branching and remodeling, resulting in the formation of liver lobes and bile ducts. Hepatocytes acquire metabolic functions through intricate regulatory networks and interactions with neighboring tissues and signaling molecules. The ventral and dorsal mesenteries are essential for the proper positioning and fixation of the gastrointestinal tract. Failure to close them correctly can lead to hernias or intestinal malrotation. Closure occurs during abdominal wall development, while the dorsal mesentery fuses and remodels. This process ensures the correct alignment and development of abdominal structures. Efficient nutrient absorption in the small intestine relies on structures called villi and crypts. Villi increase surface area for absorption, while crypts house stem cells for epithelium renewal. Their development involves complex interactions and signaling pathways currently under investigation. Meckel's diverticulum is a congenital abnormality that can occur during intestinal development. It is a remnant of the embryonic intestinal duct, which normally regresses during development. However, in some cases, the regression is incomplete, resulting in the persistence of Meckel's diverticulum. This structure can lead to complications such as intestinal obstruction, inflammation, or ulceration if not properly resolved or detected early. Understanding the mechanisms underlying Meckel's diverticulum formation can help in its diagnosis and management. During the development of the anal canal, a separation occurs between the hindgut and ectoderm, leading to the formation of an anal opening. This separation is critical for the establishment of proper gastrointestinal function and the distinction between the digestive and excretory systems. Variations in the formation of the anal canal can result in developmental anomalies, such as anal atresia or fistulas, which require surgical intervention or corrective measures. In summary, the development of the gastrointestinal tract is a complex process involving the formation of different structures. Understanding this process is crucial for diagnosing and treating gastrointestinal disorders.